Every atom in your body right now has been here before. Not here in this room, here on this planet, and before that, in the cloud of gas and dust that collapsed into this solar system 4.5 billion years ago, and before that, forged in the interior of a star that had already lived and exploded before the sun was born. The carbon in your left hand was not made on Earth. No carbon was made on Earth. It was manufactured in stellar cores operating at temperatures of 100 million degrees, scattered by supernovae across distances measured in light years, drifted for millions of years through interstellar space, captured by the gravitational collapse of this solar system, dissolved into the early ocean, absorbed into the first living cells, and passed forward through every organism in an unbroken chain until it arrived here, temporarily organized into the shape of a hand that is holding a phone or resting on a desk or doing whatever your hand is doing right now. You are not made of the Earth. You are made of the universe, and the Earth borrowed you for a while. This is not a poetic opening. It is a statement about atomic bookkeeping. Every atom that enters a living body comes from somewhere, and every atom that leaves goes somewhere. The body does not create or destroy a single one. It only rearranges them, assembles them into proteins and membranes and electrical signals and the specific configuration of matter that produces, for a finite time, the experience of being you. And then, inevitably, it disassembles them again. And what happens during that disassembly? The precise, ordered, astonishing sequence of events that unfolds when a body stops maintaining its own structure is one of the least discussed and most physically remarkable processes that occurs on the surface of this planet. This is the story of what happens to your atoms when you die. And the question you cannot yet answer is this: if no atom is ever lost, if the first law of thermodynamics holds without exception, then what exactly does it mean to say that a body disappears? Here is the paradox that physics presents when you try to think clearly about a coffin. Everything you know about death is about ending. The heartbeat ends, consciousness ends, the warmth of the body ends, the movement ends. If you were to translate these observations into a physical description, you would conclude that something has gone to zero, that a system has collapsed, that energy has been lost, that the complexity of a living organism has been replaced by nothing, or by something close enough to nothing that the difference seems negligible. That is exactly the wrong conclusion, and the error is not emotional. It is a genuine physical misreading of what a living body is and what ceases when it stops living. Here is the problem of scale that human intuition cannot resolve without help. When you observe a person alive and then observe a body at rest, you are looking at the same atoms arranged in a different configuration. The difference that feels catastrophic to you, the difference you call death, is, from the perspective of the atoms involved, a change of address, a change of molecular partnership, a redistribution of energy flows. The atoms are not diminished. They are not erased. They do not stop participating in chemical reactions. They stop participating in this particular set of chemical reactions, the ones organized by a living metabolism, and they begin participating in another set, equally complex, equally ordered, equally subject to every physical law that governed them the moment before. Your intuition was calibrated to see living bodies as fundamentally different from non-living matter. Physics does not share that intuition. Physics sees a continuous field of matter and energy, portions of which are temporarily organized into high-complexity structures we call organisms. When the structure disassembles, the matter continues, the energy continues. Only the particular local organization ends. And the question you still cannot answer is this: what exactly is the sequence? What happens first? What happens second? What happens at the scale of the molecule and the microbe and the soil? And over what timeline does the thing you call your body return to the thing you came from? To understand what happens inside a coffin, you first need to understand what a living body actually is in physical terms, not in biological terms, not in chemical terms, but in the most compressed physical description possible. A living body is a dissipative structure maintained far from thermodynamic equilibrium by a continuous flow of energy. That sentence is the razor. Everything else is elaboration. What it means is this: the universe, left to itself, moves toward equilibrium, toward the state of lowest available energy, maximum entropy, minimum organization. A warm thing in a cold room becomes cold. A structured molecule in a disordered solvent dissolves. Concentration gradients flatten. Differences disappear. This is the thermodynamic arrow of time, and it points in one direction only. But a living body does not obey this tendency, not while it is alive. It maintains its temperature above the environment. It maintains concentration gradients across membranes that would dissolve in seconds without active work. It keeps its proteins folded into precise three-dimensional configurations that require continuous energy input to sustain. It builds order continuously against the universal tendency toward disorder. It does this by consuming energy from outside itself, from food, from oxygen, and using that energy to perform the constant maintenance work that keeps the structure intact. The physicist Erwin Schrödinger called this process feeding on negative entropy. The body is not a static thing. It is a process. It is a river that maintains its shape by constantly replacing the water that flows through it. The specific molecules in your liver right now were not there 2 months ago. The calcium in your bones is continuously exchanged with the calcium dissolved in your blood. Your body is not the same collection of atoms it was 10 years ago. It is the same pattern, replicated forward in time by a process that required uninterrupted energy flow. Here is the fact that most people do not know. The human body contains approximately 7 octillion atoms. That is 7 followed by 27 zeros. 7,000, 1,000, 1,000, 1,000, 1,000, 1,000, 1,000, 1,000, 1,000. Of those, roughly 65% by mass are oxygen, 18% carbon, 10% hydrogen, 3% nitrogen, and the remaining 4% a mixture of calcium, phosphorus, potassium, sulfur, sodium, chlorine, and trace elements including iron, zinc, copper, and a dozen others. The iron in your blood, approximately 4 grams of it, enough to make a small nail, was manufactured in a red giant star that died before the Earth existed. The calcium in your skeleton was assembled in stellar nucleosynthesis over timescales your nervous system has no architecture to process. You are, in the most literal physical sense, the recycled interior of dead stars, and the common belief about what a body is could not be more wrong. The common belief is that a body is a thing, a stable, persistent object, like a stone or a table, that exists until something external destroys it. The mechanism reveals something entirely different. A body is an ongoing event. It is a performance, not a sculpture. The moment the performance stops, the moment the energy flow ceases, the structure does not simply sit there intact. It begins immediately to revert toward the thermodynamic equilibrium it was spending its entire existence resisting. The engineering analogy is this. Imagine a building whose walls are held up not by structural steel, but by a continuous internal pressure that requires constant pumping to maintain. The moment the pumps stop, the walls do not hold. The disassembly begins the instant the energy stops. Your body is that building, and it has been running its pumps every second you have been alive. Death is not an event. It is the beginning of a process, and the process has layers that unfold with the precision of a clockwork, each triggered by the cessation of the one before it, operating simultaneously at the scale of the molecule, the cell, the organ, the organism, and the surrounding environment. Within seconds of cardiac arrest, the cells of the brain begin to experience oxygen deprivation. The neurons, which consume 20% of the body's oxygen supply, despite representing only 2% of its mass, cannot sustain their electrical gradients without continuous ATP production. The sodium-potassium pumps embedded in every neural membrane, proteins that use the energy of ATP hydrolysis to maintain the electrochemical difference between the inside and outside of the cell, begin to fail. Without the pump, Sodium floods in. Potassium floods out. The membrane potential collapses. This is not a metaphor for shutdown. It is the literal equalization of a concentration gradient. The same physical process as the mixing of warm and cold water, happening at the scale of individual ions crossing a membrane 7 nanometers thick. Within minutes, this cascade reaches every cell in the body. The mitochondria, the organelles that convert oxygen and glucose into ATP, stop producing energy. Without ATP, every active transport mechanism in every cell membrane begins to fail simultaneously. The cells that depend most heavily on active transport die first. Neurons and cardiac muscle within 4 to 6 minutes, liver cells within 1 to 2 hours, skeletal muscle cells over several hours, skin cells over a day or more. The body does not die all at once. It dies in sequence, system by system, cell by cell, each one surrendering its maintained organization at the moment its energy reserves are exhausted. This is layer 1, the physics of gradient collapse. The living body was a system of maintained differences. Differences in concentration, temperature, electrical potential. Death is the equalization of those differences. The physical principle in operation is the second law of thermodynamics, not as a background abstraction, but as an active force that the body had been fighting at every membrane and every mitochondrion, and that begins winning the moment the fight stops. Layer 2 begins within hours and unfolds over days. When cells die, they release the enzymes that were contained within them, proteases, lipases, nucleases, the molecular machines that normally break down cellular debris inside specialized compartments called lysosomes. When the membrane of a lysosome ruptures, these enzymes escape into the surrounding tissue. They begin digesting the cell from the inside. This process is called autolysis, self-digestion. Every cell in a body contains the precise chemical toolkit required to dissolve itself. Under normal living conditions, that toolkit is locked in compartments and regulated by the cell's energy-dependent containment systems. When the energy stops, the locks open, and the digestion begins. This is not a foreign invasion. It is the body dismantling itself, using its own enzymes, following the same chemical pathways that were used to build it. The proteins that were assembled by ribosomes are now being disassembled by proteases. The lipids that were organized into membranes are being hydrolyzed by lipases. The DNA that was packed into the nucleus is being fragmented by nucleases. The specificity of the chemistry is extraordinary. These are not random reactions. They are the same reactions that run in digestion and cellular turnover, now operating without the regulatory controls that would normally constrain them. The body is eating itself with perfect biochemical precision. Layer 3 is the one no one anticipates, and it is the layer that reveals the true scale of what a body contains. A living human body harbors approximately 38 trillion microbial cells, roughly equal to the number of human cells, some estimates placing the ratio even higher. These microorganisms, bacteria, archaea, fungi, live primarily in the gut, but also on the skin, in the mouth, in the lungs, in every cavity and surface that contacts the outside world. During life, they are regulated. The immune system maintains boundaries. The epithelial barriers of the gut wall contain the microbial population to its designated zone. The antimicrobial peptides, secreted by skin cells, suppress microbial growth on the surface. The body and its microbiome are in a negotiated equilibrium, a managed coexistence sustained by active immune surveillance. When the body dies, the immune system stops. The barriers fail as the epithelial cells that maintain them undergo autolysis. The gut bacteria, suddenly free of the containment that kept them within the intestinal lumen, begin migrating through the gut wall into the surrounding abdominal cavity. They encounter a nutrient-rich environment of precisely the composition they are most capable of metabolizing. They begin reproducing. Within hours, what was a regulated population of 38 trillion cells becomes an uncontrolled exponential expansion. The bacteria produce gases, hydrogen sulfide, methane, ammonia, carbon dioxide, that accumulate in body cavities and give early decomposition its characteristic visual and olfactory signature. This is not rot in the casual sense. This is the transfer of the body's organic material from the biochemistry of eukaryotic cells to the biochemistry of prokaryotic cells, a shift in the organizing principle of the same atoms, from the metabolic logic of the human organism to the metabolic logic of the microbial community. The atoms do not change. The enzymes operating on them change. The energy flows change. The organizational principle changes. This is what the physicist means when they say that what ends at death is not matter or energy, but a particular configuration of information. The specific set of instructions encoded in a genome and expressed through a lifetime of cellular activity. Now, a named reference point. Arpad Vass, then at the Oak Ridge National Laboratory in Tennessee, spent years working with the Body Farm, the outdoor forensic research facility established by anthropologist William Bass at the University of Tennessee in 1981, to characterize the chemical compounds released during human decomposition. In a 2004 study published in the Journal of Forensic Sciences, Vass and colleagues identified over 400 volatile organic compounds released during the decomposition process. 400 distinct molecular species, each a product of specific biochemical reactions, each carrying information about the stage and rate of decomposition. The body does not simply fall apart. It disassembles along precise chemical pathways, releasing a specific chemical signature at each stage. A signature so consistent that it can be used to estimate time of death, identify the presence of a body beneath soil or water, and determine the environmental conditions of decomposition. 400 compounds from a single disassembling body. A pharmaceutical laboratory attempting to replicate even a fraction of this chemical complexity. Deliberately, with instruments and reagents and controlled conditions, would require years and millions in equipment. The body performs this same chemistry spontaneously, inevitably, powered by nothing but thermodynamic gradients and microbial metabolism at room temperature in the dark. The industrial comparison must include orders of magnitude. The complete chemical analysis of a human body's decomposition products would represent roughly 500,000 person-hours of laboratory work using current analytical methods. The decomposition itself takes weeks. The chemistry is not simplified by the absence of life. It becomes, in some respects, more chemically complex because the tightly regulated metabolic pathways of the living organism are replaced by the overlapping, competitive, unregulated chemistry of dozens of microbial species simultaneously consuming the same substrate. This is not the chemistry of simplification. It is the chemistry of redistribution. If you subscribed to this channel and clicked the notification bell, you would have known this video was coming. Subscribe now and click like. It tells the algorithm that people actually want to think, and that makes these videos possible. Here is what the chronic absence of this understanding costs, not emotionally, but physically, in terms of what we do with bodies and what that does to the systems that depend on decomposition. The modern approach to death in industrialized societies involves embalming, the injection of formaldehyde-based preservative solutions into the circulatory system to halt decomposition, followed by burial in sealed metal or hardwood coffins, inside concrete burial vaults, in manicured lawns managed with herbicides and pesticides. The intention is preservation. The physical consequence is the removal of a body from the decomposition cycle that the surrounding ecosystem depends on. Consider the time scale and compare it to something familiar. The natural decomposition of an unembalmed, unenclosed body in temperate soil takes between 1 and 5 years to reach the stage of skeletonization, and between 15 and 30 years for the organic components of the skeleton itself to fully reintegrate into the soil matrix. That is a process operating on the timescale of a human generation. A sealed, embalmed body in a concrete vault has been documented to persist with minimal soft tissue decomposition for decades. In some cases, over a century. The embalming slows microbial activity. The sealed coffin prevents oxygen exchange with the soil. The vault prevents direct contact with the biological community of the soil altogether. This is the entropy problem framed precisely. A system that has been removed from the dissipative cycle persists in a state of arrested organization. The atoms are locked in configuration. The energy that could be released into the surrounding soil ecosystem, the nitrogen, the phosphorus, the carbon, is withheld. The soil microbial community that would normally process a decomposing body receives nothing. The mycorrhizal networks that connect plant roots and depend on decomposing organic matter for a portion of their nutrient input are deprived of a source. Compare this to the familiar physical process of a tree falling in a forest. A fallen tree in an intact forest ecosystem is processed by fungi, bacteria, wood-boring insects, and small vertebrates over approximately 20 to 80 years, depending on species and climate. During that time, it releases its carbon and nutrients back into the living system, absorbed by the roots of neighboring trees, incorporated into fungal networks, passed up through food chains. Remove the tree by combustion or encasement, and the local ecosystem receives a gap in its nutrient budget. Multiply that gap by the scale of modern cemetery practice, approximately 2.7 million deaths per year in the United States alone, the majority processed through embalming and sealed burial, and the aggregate effect on local soil ecology is measurable. Jennifer De Bruyne at the University of Tennessee has studied the soil microbiome around decomposing human remains since the mid-2010s, and her findings are not ambiguous. The microbial community in soil immediately beneath a decomposing body undergoes a profound transformation, with the proliferation of specific bacterial species capable of processing the complex organic compounds released by decomposition. The zone of soil affected by a single decomposing body can extend meters in radius. This is not a contamination. It is an enrichment, the incorporation of a body's atomic content into the living matrix of the soil, the passage of organized matter back into the nutrient cycle from which it came. What you have been calling the end of a life is, in physical terms, the beginning of its most productive ecological phase. The common belief must be reformulated. We have been taught to think of a body after death as waste, something to be managed, contained, preserved against the indignity of dissolution. The mechanism reveals something different. A body after death is a concentrated deposit of biological capital. Nitrogen, phosphorus, carbon, and dozens of trace elements in organic forms that the soil community is specifically equipped to process. Withholding it from that community is the equivalent of burning a library and calling it preservation. This is not an argument about funeral practice. It is a statement about what a body physically is and what physical process its atoms are scheduled by thermodynamics and ecology and 4 billion years of evolutionary history to participate in. The gesture this series studies is always the smallest possible. The blink, the breath, the glance upward, the hum in the throat. This script is different in one respect. The gesture it studies is not performed by the living. It is performed by the body itself, without instruction, without conscious participation, at the moment the living organism stops providing instructions. But that difference is smaller than it appears, because the same atoms that will eventually participate in the decomposition process are participating right now, in you, in this moment, in processes of equal or greater complexity. The carbon in your exhaled breath was, minutes ago, a glucose molecule in your bloodstream. Before that, it was a starch in food you ate. Before that, it was carbon dioxide absorbed from the air by a plant. Before that, it was exhaled by another animal. Before that, it was in the soil. Before that, it was in the sea. And before all of that, billions of years before, it was forged in the nuclear furnace of a star. Your breath, right now, is stellar exhaust. Here is what the body system that seems furthest from decomposition actually shares with it. Your gut microbiome, the 38 trillion organisms that live in your intestine, is performing right now the same basic biochemical operations that will occur post-mortem: fermentation, proteolysis, lipolysis, gas production. The difference between the microbial activity inside your living gut and the microbial activity inside a decomposing body is not a difference in chemistry. It is a difference in regulation. You are right now managed decomposition. Your immune system and your epithelial barriers are the control rods that prevent the reaction from becoming uncontrolled. Death is not the introduction of a foreign process. It is the removal of the regulatory machinery that keeps a process you already contain from running without constraint. This mechanism has been present for as long as multicellular life has existed, approximately 600 million years. Every multicellular organism that has ever lived on this planet has contained within it both the metabolic machinery for life and the microbial and enzymatic machinery for its own dissolution. Evolution did not eliminate the dissolution machinery. It built regulatory systems on top of it. Because dissolution is not the enemy of life at the ecological scale. Dissolution is the precondition of life. Every atom that enters a living body had to be released by a previous living body first. There is no life without prior death. There is no carbon available to new organisms without the decomposition of old ones. Modernity's relationship with the body's dissolution machinery is the relationship of someone who has covered the exhaust pipe of a system they depend on for air. We have built a civilization that extracts extraordinary complexity from the natural world, and then, at the point of return, the point where the atoms should flow back, we interrupt the cycle. We embalm and encase and seal, not because of physical necessity, but because of a cultural belief that the dissolution of a body is an insult to the person it belonged to. The physics does not recognize that distinction. The atoms do not recognize that distinction. And the ecosystems that are waiting for those atoms, patiently, with the biochemical patience of 4 billion years of evolution, do not recognize that distinction either. If you want to see another video in this series, subscribing takes 2 seconds, and it actually helps. The algorithm learns that depth matters to people who watch here. Like it too. It costs nothing, and it changes what gets recommended next. So, what happens, precisely and in physical terms, to your atoms inside a coffin? In a sealed, embalmed, concrete-vaulted coffin under the conditions of standard modern burial in a temperate climate, the following sequence occurs with variations based on embalming quality, coffin integrity, and soil conditions. In the first weeks, the embalming preservatives, primarily formaldehyde, which cross-links proteins and arrests microbial activity by binding to amino groups in biological molecules, suppress the autolytic and putrefactive processes that would otherwise begin within hours. The body exists in a state of chemical suspension, not preservation in the true sense. The formaldehyde itself is metabolized, and its concentration declines over months, but a significant delay in the onset of active decomposition. The body in this phase looks, externally, relatively intact. Internally, without the energy of the living metabolism to maintain cellular gradients, the cells are all dead. The structure is held together by the cross-linked protein matrix that formaldehyde created. Between weeks and months, as the formaldehyde concentration drops, microbial activity begins to resume. The microorganisms that survived embalming, primarily spore-forming bacteria capable of withstanding formaldehyde, begin to reproduce. But in a sealed coffin without oxygen exchange, the dominant metabolism is anaerobic. The bacteria that thrive are not the aerobic decomposers of open-air taphonomy, but anaerobic fermenters and methanogens. Their metabolic products, methane, carbon dioxide, hydrogen sulfide, ammonia, and a range of organic acids, accumulate in the sealed space. The chemistry of decomposition inside a sealed coffin is significantly different from the chemistry of decomposition in open soil. The products are the same elements, but in different molecular combinations, and they accumulate rather than dissipating into the surrounding environment. Over years to decades, the soft tissue gradually breaks down even in the sealed environment. The proteins hydrolyze, the lipids oxidize or saponify, in some cases forming a waxy, soap-like substance called adipocere, a calcium soap made from the fatty acids of subcutaneous fat, which can persist for centuries under the right conditions. Adipocere is one of the more extraordinary physical transformations in decomposition. The same fat that provided thermal insulation in life is converted, by microbial and chemical action, into a chemically stable solid that holds the general form of the tissue it replaced. A body transformed by adipocare can retain its external shape for hundreds of years, while the soft tissue it represents has been entirely chemically reorganized. The bones, depending on soil chemistry and coffin integrity, may persist for centuries to millennia. Bone is composed of approximately 30% organic material, primarily collagen, and 70% mineral, primarily hydroxyapatite, a crystalline calcium phosphate. The organic fraction degrades over decades. The mineral fraction persists until soil chemistry dissolves it, a process that may require hundreds to thousands of years in alkaline soils, or as few as decades in highly acidic conditions. The minimum effective dose of this knowledge for changing your physical relationship to the body you currently inhabit is this: pay attention to what you do with your gut microbiome. The same organisms, the same biochemistry, the same molecular logic. Everything that is happening to a decomposing body in the soil is happening in regulated form inside you right now. What you feed those organisms, what substrates you provide for their metabolism, determines their composition, their byproducts, their effect on your immune system, your nervous system, your hormonal state. This is not a metaphor for the connection between life and death. It is the literal mechanism by which the chemistry of decomposition and the chemistry of life share the same molecular cast of characters. The variation for those who find this uncomfortable: you do not need to think about decomposition at all. You need only to think about the soil. The soil is the destination of the atoms currently organized into you. Whatever improves the health of the soil, organic material, microbial diversity, undisturbed fungal networks, is what your atoms will return to. Tending the soil is, in a physically precise sense, tending the system that will eventually incorporate what you are made of. If that strikes you as either morbid or comforting, depends entirely on what you believe a body is. If you believe a body is you, it is morbid. If you believe a body is a temporary configuration of atoms borrowed from a planetary cycle, it is simply accurate. What you should observe as a signal that this mechanism is legible to you: the next time you hold soil in your hand, notice that you are holding the processed remains of every organism that has ever lived and died in that location. You are holding the output of billions of years of decomposition chemistry. The soil is not dirt. It is the accumulated product of exactly the process that will eventually apply to you. The error that nullifies understanding is the belief that death is chemical inactivity. The opposite is true. Death initiates one of the most chemically active processes a body undergoes. The living body is, in thermodynamic terms, a suppression of chemistry. Death is the release of that chemistry. What the coffin does is slow and alter, but not prevent, the inevitable return of organized matter to its constituent elements. The thermodynamic gradient that was maintained by the living organism disappears the moment the organism stops living, and the universe begins, immediately and without ceremony, the work of equalization. Before the last block, one question for you, and it deserves a real answer, not a yes or no. When you think about what your body is made of, the carbon, the iron, the calcium, do you actually feel any different about where those atoms came from? Or does the stellar origin still feel like an abstraction that the body's warmth prevents from landing? The last time physics was asked to account for death, it did something unexpected. It refused to recognize the category. The equations that describe the motion of matter and the flow of energy do not contain a term for death. They contain terms for mass, velocity, temperature, entropy, potential, kinetic energy, electromagnetic field. They describe the behavior of atoms under every condition: inside a star, inside a cell, inside a coffin, inside soil, inside the atmosphere. They are continuous across what we call the boundary between life and death, because from the perspective of the equations, there is no boundary. There is only the continuous rearrangement of the same matter according to the same laws. This is not a way of saying that death does not matter. It is a way of saying that what we call death is a human category imposed on a physical process that does not pause for categories. the atoms that will eventually disperse from a body into soil and water and air, and eventually into other organisms, and eventually into the atmosphere, and eventually, over geological time, back into the mineral cycle of the planet. Those atoms are not liberated by death. They were never imprisoned. They were organized, temporarily, into a pattern complex enough to experience itself. And that experience, the experience of being a particular arrangement of stellar material that learned to ask questions about itself, is not diminished by its temporariness. It is made possible by it. The carbon that is in your body right now will, over the next few billion years, pass through dozens of other organisms, dissolve into the ocean, precipitate as limestone, subduct into the mantle, be released by a volcano, be absorbed by a plant, be eaten, be exhaled, be captured again. It will continue participating in chemistry for as long as the Earth has chemistry to offer. And the chemistry it participates in will always be more complex and more astonishing than you would expect from something as simple as a carbon atom. Because complexity is what matter does when you give it enough time and enough energy and enough variance in conditions. You are what carbon looks like after 4 billion years of trying. The body that is sitting or standing or lying somewhere. Reading these words is the current address of atoms that have been in continuous circulation since before the planet existed. The dissipative structure that runs on those atoms, the self-maintaining, self-repairing, entropy-resisting, question-asking pattern that you call yourself, is not a thing that was built and will be destroyed. It is a process that has been running, has reached this moment of self-awareness, and will eventually hand its materials back to the process that lent them. The coffin is not the end of the story. It is one possible penultimate chapter before the atoms disperse back into the cycle they were always part of. Physics does not grieve this. Physics simply continues the calculation. And the calculation, followed to its conclusion, arrives at something that feels less like comfort and more like precision. You are not a visitor to this planet who will someday leave. You are a temporary local organization of the planet itself, assembled from its materials, powered by its star, governed by its chemistry, and scheduled by the same laws that assembled you to return. The universe does not lose you when you die. It only changes what it is doing with your atoms.